שני חלקים, בחלק העברי כמה משפטים. אין הרבה הזדמנויות שיש לנו אירוע מהסוג הזה, והרבה פעמים תהיתי מה לנו, המתרגמים האלה שמנסים להביא קהילת העיתונאים לשערנו, לקהילה המדעית. אין ספק שזה קריטי. לא יכולתי לבחור יום טוב יותר להסביר למה התקשורת המדעית חשובה. מי שראה את עיתון הארץ הבוקר על לימודי ליבת מורת הכרה בתי ספר יכול להבין כמה חשוב בהחלט להכניס את המדע לחברה, ומשום מה יש תחושה בעולם המערבי שכאילו העולם המערבי, המדינות המפותחות מחוסן, אבל השאלה של דרוויניזם גם בארצות הברית וגם במדינות אירופה עולה מחדש. אנחנו עם הקהילה המדעית שבאה לכאן שותפים למאמץ אחד גדול, גידול, בירור הבערות, הבטחה של מעבר של אינפורמציה, ולימד אותי הלמוט שוורץ, ידיד שלנו מגרמניה, הוא אמר, Science is trust building. המדע חשוב בבניית אמון לתקשורת. זה מה שמאחד את כולנו כאן, ולכן חשוב בכל מדינה, בכל מסגרת, אנחנו חייבים גב, אנחנו קטנים מדי בארץ הזאת, ואנחנו צריכים את העזרה של הפעילות העולמית, והפעילות העולמית צריכה אותנו. הנחה ראשונה. הנחה שנייה, ואני אומר את זה לידידינו העיתונאים שבאו, במהלך כל תקופת עבודתי והיכרותי מהמערכת המדעית, כל השנים, הקהילה המדעית במדינת ישראל הושיטה יד וניהלה או באופן אישי או באופן מוסדי קשר עם שכנינו באזור. גם בימים אלה, שהמצב נראה אביב עמים, לא יציב, גם בימים אלה אנחנו מקיימים פעילות אינדיבידואלית ומוסדית עם השכנים שלנו, והסיור בססמה יחד איתם הוא כדי להראות עד כמה כנה ונכונה ואמיתי הוא הרצון שלנו לבנות קשר עם השכנים. מקובל לומר תמיד שתיבת פנדורה, כשפותחים את תיבת פנדורה יוצאים רק דברים רעים. מי שמכיר את הסיפור, את המיתולוגיה היוונית, בסוף הקופסה, בתחתית הקופסה, ישנה גם התקווה. בתוך תיבת פנדורה, המדע הוא התקווה, ואנחנו ננסה לעשות את זה ביחד. I tried to turn to English. This part, I think I'm going to introduce Sascha, so to make it easier, I think we should be the group that managed to come to this peaceful part of the world, the Middle East. We are very happy to have you here, really very happy, and we want to gather forces with you. The point, I think, is that we have to find a kind of nice nickname for the group. So I was thinking of having since there are only 12 at the minute, at the moment, the Magnificent 12, but, you know, in Hebrew, I think it's better to call you, because of one good reason. Our colleague from Egypt couldn't make it. He got a visa from us, but he couldn't get the visa from his own authorities. So I think we should call this group the Bar Mitzvah team. So the Bar Mitzvah team will certainly mean maturity, change in the whole atmosphere, and I hope we'll take it in the future more seriously, anything that has to do with science communication. Enough of this part, kind of introduction, and I would like to ask now to turn to the program, and I'd like to ask Sascha Karberg, who is the co-founder of the German Science Journalism Network. I have to be careful now because he taught me how to pronounce it. Schnittstelle, and the former fellow at the Night Science Journalism program at MIT, to come and give us his talk, please. He was awarded a number of awards in journalism. I'm not going to mention all of them. And he is writing mainly in life sciences in the international and German newspapers. And Sascha, permit you to use your first name after a short introduction earlier this morning, is going to talk about experimental science journalism, how 4 journalists became biohackers to report about the growing DIYbio-movement. Thank you, תודה רבה.
Yeah, thank you for the kind introduction. Thanks to all the organizers, Eilert, Karen, Avital, I think so many were involved. I can't name all of them, and it's really a great opportunity to be in Israel, and it's my first real visit. And yeah, now I will present a journalistic expedition that I did together with 3 of my colleagues. An expedition into a wild jungle, an urban jungle. We found and described a new species, the biohackers. Some call them also biopunks or amateur geneticists. Yeah, lots of names. Outlaw biologists is another term. They don't like that actually. So, and on the course of this journey, we kind of self-evolved into biohackers in a kind of a Kafka way. And I thought I should give you a little glimpse in there, like cadmium or whatever. And so this, and they developed kind of an enthusiasm because these students before that they just learned theoretically about genetic engineering, and this was the first time they were able to actually do something with their knowledge. So within a very short period, they organized everything. Sometimes they organized their own lab at the university. They organized a professor who should supervise this. They did more or less everything. Sorry, they did. They did everything by themselves, and this enthusiasm of open source idea also, where they put genes together, this kind of left the university, because when these students left the university for some reason, or when they get back to the classes or so, they wanted to go on with their genetic engineering. And so they built their own labs outside of the university. At home first, later then in so-called hacker spaces, biohacker spaces, and came together with amateurs from other fields and started this biohacking movement. And this happened a few years before I was at MIT, and I got involved with these people. I spoke with them. That's what you do during this fellowship. And the first I visited was K.O. She is probably really one of the very first biohackers. She checked her own genes for a certain disease called hemochromatosis, and because her father was diagnosed with that and couldn't really understand the papers, and her idea was not to check her own DNA just by giving it to a physician, but do it on her own, with her own possibilities at home. And she actually has a closet in her bedroom, which is full of equipment like PCR and machines and pipettes and stuff like that. So it's some of the pictures are in the movie, but I can't show everything to you. So, and she started a little community there together with others, Meg Howell and Jason Bobe, and they founded a website, DIYbio.org. Enough, at least, to write an article about that. That's what I did in 2009. Yes, it was published in 2009, but I wasn't really satisfied with that. And for a reason, because when you go out as a science journalist, you usually want to talk with experts, appropriate experts in the field. You check this with published studies at PubMed or other sources. So you read these studies and so on. But here were no studies. You couldn't read anything about biohacking, mutual information or so. You just could talk to these biohackers, and of course they told you, we can do everything, we can change the world, we will do that. And then you go to scientists and they say, what? Biohackers? Never heard of that. So what can they achieve? They can't achieve anything. because you need a professional lab for that, and despite all this, there might be a safety and security issue with that, and stuff like that. So, but these were only guesses, because these professors usually, at least in Germany, and some of them also in the US, never saw a biohacking lab from the inside. So I was kind of forced to write the story around these guesses and speculations, and was not really satisfied with that. So I came to the idea, why not do it yourself? Because you were trained as a molecular biologist. Okay, that was kind of some years ago. So, and I was not very keen to do pipetting again, because it's really a tedious work, and some of you might know that. So, but and also the editors thought, that's one email said, that's a nice idea, but a little bit too much work, right? And also expensive, of course, because the equipment that we finally bought summed up to about 3,500 euro. And but we were lucky, we found grants for that, because there were research fellowships in Germany. Yeah, and also we got a bigger grant for making a 60-minute documentary about that. So that's how I ended up on the screen. So because we just sent this idea there, and usually only cultural journalism projects win this, and I think we were the first science-related project. So yeah, then you have to do a 60-minute TV documentary. So this kind of grew into a bigger project. So we bought a PCR machine. You can see it over there, the very big thing. Okay, so in the very beginning, I remembered during my very first studies at the university, we were standing on such a machine, and at the time, I have to admit, I didn't really understand how to really put the program in, because it took us about 3 days to find it out, because you can't find the manuals on the internet anymore. So it really took a while, and so the cost at that time was about 10,000 euro, and the professor was always running around us, don't touch it too much, don't touch it too much. And but now it's about 250 euro at the internet, eBay. Okay, so and all the other equipment we bought on the internet or at the local stores, because you usually can just take regular stuff. You don't have to use the high-end research solutions or chemistry. So as you can see, I don't always wear stuff here, so, and it's sometimes hot in Berlin too. So yeah, we started not only the lab, but we also got in contact with these biohackers, and it was a very big advantage not to be a journalist who only wanted to write up a short story about them and just wanted to criticize or just pick up the kind of horror scenario, like some people doing genetic engineering in their garages or so. We really talked with them sometimes about protocols. Actually, how are you doing this, and stuff like that. So we were on a completely different level of speaking with them. So they were open-minded. We visited first, of course, the KO in Boston. Below that, here on the left side, is Jason Pope. He is also a Harvard professor, and is very much involved in setting up a landscape around biohacking. For example, he invented Ask a Biosafety Officer thing on the website. So biohackers all over the world can put their questions there and ask biosafety experts from the US, should I do that or shouldn't I do that? And that's actually really helpful, because sometimes you run into these questions if you want to make an experiment at home or in a biohacker space, and you don't have the facility. You can't just go to the next lab and ask, did you ever do that? Can you help me with that? So biohackers, they might be forced to try it at home just by chance, and might do some mistakes, like that. These things happen, of course. So he found a way now to finance this, and because the biggest problem was insurance, of course. So no insurance company really wanted to carry the risk of the safety officers that they might say something wrong or so, and something happens because of their advice or so. But Lloyd's usually makes this for a huge amount of money, I think. So then also we found in San Francisco, we talked to people who were actually just starting to build a biohacking lab right in the middle. On top in the middle is the lab in New York, Brooklyn, GenSpace. I think they are the most advanced. We talked to Steve Kurtz here on the left. He was the first so-called biohacker. He's actually a bioartist, but he used genetic modification, and the FBI heard about that and kind of arrested him for a few days, and he really got in trouble. But the FBI, meanwhile, they learned a lot. They actually invited biohackers from all over the world to a so-called outreach workshop in Walnut Creek, and I've been there too, and it was really a creepy experience, because all these agents were running around, and they were kind of friendly talking to you, what they usually don't do, I think. And it was they wanted to just inform the community about what might be difficult. So and also they wanted to learn how to distinguish between a drug lab, for example, where someone's doing amphetamine or whatever, and a biohacking lab. They look different, but not for the regular policeman, of course. So it's a lab in the end. So they wanted to provide guidance for these. So, for example, in the establishment of the GenSpace lab in New York, they were involved by making the right connections to the fire department and stuff like that, so that all the regulations were in place. So they have a proactive approach, and if you talk to them, they don't see a real threat in this movement, because the people who are working there are usually biologists, because if I talk to them, their background is highly educated, at least. Even if they are not biologists by training, they usually have an academic background, so they are familiar with safety regulations and also security. So, yeah, I shouldn't stay too long. So, but as I said, not on just talking, we had to go back to the lab too. And it took much longer than we thought, and if I would have known that in the very beginning, I would have never done that, because it took really months, and I stood nights in the office, and you can here you can see that this is an open office, okay? So these are not journalists who are sitting there in the back. These are architects, and they sometimes look through the window, what are these guys actually doing? And so we tried to explain, but I don't really believe that they understood it so well. But okay, so what did we really do? So one of the experiments was, a smelly one. We picked up dog poo, which is famous for Berlin, I would say. So I think sometimes there are more dogs than people, and so you can extract the DNA from there, and you can also extract the DNA from the saliva of the dogs. That if you throw little balls for them, and then you make a DNA fingerprint, as everyone knows from CSI and so on. And there were a lot of mistakes, and I wouldn't really trust the end, the results or so. But it was a nice idea to think about that this is already possible with a very easy equipment. So you don't need to have a fancy CSI lab for that. You can do this DNA fingerprint, of course, also with the human DNA, okay? And everyone might be able to think of some possibilities where this might be useful. So talking about girlfriends and boyfriends and stuff like that. So of course we were not allowed to do this in Germany because the law in Germany is very strict about that. So you couldn't just take DNA from someone else and analyze it in any way. So at least not from a human, but from a dog, they didn't write this in the law actually. So that's why we just to make clear the principle. So we also tested sushi just for checking, is it really tuna or do they sell me something else here? Whale or whatever. Actually they found whale as high school class, talking about amateurs, high school class in New York did this, and I think in San Francisco too, and I'm not sure whether the New York guys or the San Francisco guys found whale for some reason, I don't know. So but we also wanted to push the boundaries a little bit. So everyone thinks about, when I tell you about biohackers, the first thing that comes to your mind is bioterrorism. Might this be a possibility that people cook up new dangerous bacteria in their basements or so? And we wanted to check whether this is possible. So there is, I have to tell you that if you want to get dangerous DNA sequences, like for proteins like ricin. It's a potent protein. If you ingest just a tiny bit of ricin, then humans will very surely die of that. So if you want to order the DNA sequence of the gene at a company, these DNA sequencing companies, they won't send it to you because they check what you request, and of course they check to which institution you belong. So, and if you have no institution at all, just your private address, you won't get most of the DNA sequences. But and this sounds like a great safety net to keep this technology in academia and in industry and nowhere else, and not in amateur hands. But it's just not true, because the ricin gene is out there all over the place. You can order the ricin plant from the internet. You can go to the next flower shop and buy the plants, and you can go to your botanical garden, and that's what we did. We just went to the botanical garden. Okay, the picture is a little bit blurry. Yeah, sorry. And we took just a part of a leaf and extracted the ricin gene from there. So we took the DNA from the plant, and if you just order the tiny primers that say the amplification process where it should start and where it should end, then you can just amplify the gene. That's what we did. So just to tell, if someone really wants to get these genes, then you can get the genes. That's not a problem. But now comes the big but, because you have the problem that you need, if you want to do something more with that, implant this gene into that you just synthesized by yourself into a bacteria, then you won't. Then you really need higher equipment to protect yourself and to protect others, and to be really able to invent something new. And by the way, you could just use the seeds of the ricin plant to make the toxin. So you don't even need to make all this genetic engineering, and that's what you saw in the trailer. That was one professor, Professor Eckhard Wimmer, a German actually. He synthesized the polio virus, and it was a crucial experiment, but you can also find this virus out there all the time, and you find a lot of very dangerous bacteria and viruses out there that you can just take without any genetic engineering and be a terrorist. So our conclusion at the end was that the mix-up of biohackers and bioterrorists is just not sufficient. There is no strict relations. So so we published the stuff in at the Frankfurter Allgemeine Sonntagszeitung, 4 pages long. We also wrote a book about it. You saw part of the TV documentary and a 3-part series at the BBC Future website. What did we learn? I think I skipped this, what we learned, because I already told you a lot, but this is one of your pictures. I thought I should include this, because perhaps one back. Should I go back? Previous, yes, because the interesting thing about that is that, as I said, a lot of biologists are actually working there. Professional biologists are going in biohacking labs to work there, just because they can do projects there where they never get a grant for. And that's really a great idea. And that was what actually also happened in the computer hacking movement, that people did side projects there that they couldn't get funded in the regular academic career. And this little picture, I think, tells you about the difficulties of this professional career. So, I think that biohacking is, I learned that we can learn a lot from the biohacking movement to bring science to the people, and because it's another way to make science more involved in the discussion, to get people involved there. Give me a second, I'm trying to finish this here. Sorry. It's a little bit 2 computers, that's too much for me. So, yeah, I think that science journalism in general should include the perspective of the people, and I just close here and hope that I don't find my last words. So thank you. Sorry. No. So.
But would you mind taking 3 questions?
Yes, of course.
Okay, so do we have any questions? Yes.
Sorry, your last sentence said that this biohacking is a possibility for the public to be more involved. That's what you were saying in the last sentence? The opportunity.
Yes, I think that as science journalists, we usually come from a scientific background, like me. So I studied biology and so on, and I'm very much in the academic thinking also. But sometimes the patient out there, for example, he doesn't come from a scientific background. He is not really interested about the news of the new breakthrough in cancer therapy or so, which has been publicized by I know, Weizmann Institute or wherever or so, because he is interested. Can I get this therapy or not? And why do I not get this therapy? Why is it not appropriate for me? Or are there bureaucratic hurdles that I don't get this? So the process, I think the people out there are much more interested in the process of how science is implemented in their life, okay? And they want to talk about this implementation too. They want to discuss it. They want to discuss about what's really the use of the technology. So the biohackers, for example, if you talk to them, a lot of them are biologists, but they also think differently about genetic engineering. They don't agree with the Monsanto kind of way to introduce genetic engineering into the society, because it's a monopolistic view of how to use this technology, and these biohackers might have a completely different mindset, and they are independent, and sometimes, in my view, more independent than actually scientists who are involved in all this. Sometimes they get money from the industry. They are, at least people out there don't really think that scientists don't have an agenda or so, and are totally neutral. That's not the case, I think.
So they would be searching for patient will be searching for a biohacker to help him? How exactly this? Because you said most of the people are biologists anyway, so just this connection between another sentence about that.
No, I think that biohackers just raise other questions than scientists or industry or so. It's not that they might come up with new medicine. I think that's far too early to tell that. But there are biohackers out there who have ideas of treatments, different treatments than in the usual. But every one of us knows that this takes a long time, a lot of money, and so on. So sometimes these are just dreams or visions. But it's nothing bad about dreams and visions that you can't get out in the normal academic area. So there is a new area now where ideas might be tested. First tests or so, and these people might have a different view and so on. So I came out of this research quite positive about this movement. So more positive than before, actually.
We have time to one final question.
Do you think this project represents the wider field of science getting out of universities to the general public, pretty much like the internet and blogging? Some of the resources and energies of the media, traditional media, you see it going, spreading wider to other fields of science, affecting the whole university system?
You mean the general amateur scientist movement or so? I personally think that...
Maybe you can repeat the question.
I think the question is whether there will be more amateur scientists out there within the next years. Yes.
Out of the traditional system.
Yeah. Actually, you have there are also already journals about citizen science out there. So and from different areas too. So we always think all the people out of academia are lay people or so, but that's really not true. There are so many people out there who have their own terraria and have great knowledge how to raise a certain species or so. I my own know 5 or 6 people in Berlin who are really good with that. So and there are all sorts of fields. All these people who have technical skills or so. They are in these hacker spaces for years already. So and this knowledge now connects because of the internet. Okay, so these people were always very separated, but now they get together and build websites and exchange information much quicker. And this is a different setting, yes, but it's also science. And by the way, science, the professional science, started as amateur science. Every one of us knows that because one of the big discoveries were made by amateur scientists.
I would like